Hello everybody. Now today we're going to do the chapter on inherited change, and this is the video number 3. This is on the chapter which is the chapter 16 of the A2 syllabus, and in A2 biology the syllabus code is 9700, and we've done 2 videos before this, and this is the 3rd video now that we're going to talk more about the real genetics now. The first thing I want to clarify is that you do not have to know which traits are dominant and which traits are recessive. Usually in the question we're going to give you a clue. Like for instance, if I give you a clue in the question is that blue eyes is a recessive trait. Well, first of all, you've got to decide which letter you're going to choose for it. So I, looking at brown and blue, would also of course use the letter small b. So if I have, this will be of course in the question that you know what is the recessive and what is the dominant trait. So if I have said in the question that the blue eyes is the recessive trait, then you're very sure that you're going to, number one, give it a letter. If I haven't given it a letter, but usually we do give you a letter. So if I've said this, then it means the blue-eyed people are this. Why? Because you have to have a pair of alleles. You have to have a pair of the, like on a chromosome, whatever chromosome number it is. If I say this is eye color, then this person with blue eyes has a small b here and a small b here. And the small b means that there must be a certain sequence of bases. And this could be pretty long. And this sequence of bases results in the formation of a protein which has this shape and which results in the blue pigment being deposited in the iris of the eye. So first of all, you've got to understand is if I say this is the gene for eye color, so that gene must be on some, it must be a portion of a chromosome. Which chromosome? We have now actually mapped all of the human genome, so we can actually know which chromosome has the eye color gene. So, I mean, this information will be given to you in the question. So, don't sort of start worrying about, well, do you need to memorize it or do we need to really learn all this? No, I'm sorry, you don't have to learn anything. Now, I must have written in the question that brown eyes is this. Now, if I have written the capital letter, that means this is the dominant trait. And the small b is the recessive trait. So, it means people with brown eyes, and if I've given this clue in the question, then it means people with brown eyes are either this or this. They can't be anything else. Now this is called homozygous. This is called homozygous dominant. Why? Because both are capital letters. And the capital letter usually shows a dominant trait. Now the definition which is given in your books on the dominant characteristic, it says allele is one. The dominant allele is one whose effect on the phenotype of a heterozygote is identical to this effect as a homozygote. So if it's a homozygous, that effect is the same, and if it's a heterozygous, even that effect is the same. So homozygous and heterozygous. Heterozygous, please remember, is not dominant or recessive. There is no such thing, because heterozygous is only heterozygous. It can be a capital A or a small a. It can be a capital D and a small d. So heterozygous means one dominant trait and one recessive trait. And the fact that this dominant trait shows its effect, that means this one, the small b, its effect is not even considered. It is just ignored. Like we say, okay, let's go for coffee, and you say, well, let's go to Gloria Jeans, and somebody said, let's go to Starbucks, and then we ultimately go to Gloria Jeans. So, well, actually the other person said something, but we've ignored the other person. The suggestion of the other person has been ignored. So similarly, the small b, its effect is not going to show. So the big B is going to show its effect. In this, of course, we've got both big B's. In the heterozygous state, the small b is not, it's ignored. So what you need to understand is, please do not try to remember which trait is recessive, which trait is dominant. We usually give you that, except in certain diseases which we're going to teach you at the end. in which you would be told which disease is a dominant disease and which one is a recessive disease. So people with blue eyes will all be, now what is the name of this? When it's small b, we always say it is homozygous recessive. Homozygous recessive. And homozygous recessive means they are both small letters. Both small letters. So if you look at, if I say big B, or I say big A, any trait, or I say big D. Now these are all homozygous dominant. Both are capital letters. But if I say small b, if I say small a, if I say small d, now these are all homozygous recessive. These are all homozygous recessive. Right? Now, what is left? Now we have big B, small b. We have big A, small a. We have big D, small d. Now, these are all called heterozygous. And please remember, there's no, as I said earlier, it's just heterozygous. It's not dominant or recessive. So, everybody has to be very clear on this, that if we say two capital letters, then it's homozygous dominant. If we say two small letters, then it's homozygous recessive. It's one capital letter and one small letter. So that, of course, means it's heterozygous. Now, this would be given in the question. Let's say, now let's talk about the fur color of rabbit. So if I say, okay, black is the black furred rabbits. So I tell you this in the question. And the recessive trait is the white fur rabbits. Then of course we don't have any other problem. We're not going to talk about this. We're going to just say there's a dominant trait and there's a recessive trait. So if a black rabbit, now this of course would be given in the question. So black furred rabbits would be this, would be either this or would be this. And the white furred rabbits would be this. Because I've told you this is a recessive trait. So please read the question carefully. This, of course, letters you know is the genotype. And this fact that we say this is a white fur rabbit is the physical features. So pH, physical pH, phenotype. So phenotype. That's how I make you all remember it is physical features, but you can just say appearances also. So, dominant, recessive, phenotype, genotype. Now let's look at what is the F1 generation. The F1 generation is when you cross one homozygous dominant, say with rabbits, and we're crossing homozygous dominant rabbits with homozygous recessive. So this is homozygous dominant, this is homozygous recessive. Now all the offsprings are heterozygous. So this heterozygous is the F1 generation. So all the heterozygous will be considered the F1 generation. Now when we cross these, the F1 generation, so the F2 generation is the offspring resulting from a cross between two F1 heterozygous organisms. So if we are going to take these, then we're going to have, and we're going to cross these. So this is this one. This is the one, these are the ones which are all from the F1 generation. So if we cross them, then we have this possibility. So we have this, then we have this. So that's also the same. Then we have this. So it's big B, small b again. And then a small b. So this is a probability of having a black furred rabbit. And this will of course be a white furred rabbit. So the phenotype ratio is 3:1. right? So the phenotype means physical features. So there are 3 blacks and there's 1 white. So 3:1. This would be the phenotype ratio. Because when you're looking at them, we have 3 blacks and 1 white. But what will be the genotype ratio? Genotype ratio will be 1:2:1. So the genotype ratio will be 1:2:1. In the same question, these 3, the phenotype ratio will be 3:1. So genotype ratio is what is the genotype. So this one would be a big B, so 1 type, then big B, small b, 2 types, and then small b is a separate one. So we have 1, this is this one, and then this 2, and then this one. So please everybody understand what is the F1 generation and what is the F2 generation. Now let's look at the codominant phenomena. Codominant means there are 2 traits, and when they're found together, they're both codominant. Like for instance, if I say, okay, let me give you an example. Red flowers are crossed with white flowers, and all the offsprings are pink. All the offsprings are pink. So we have pink. All the offsprings are pink. So now you have a red and a white showing its effect, and of course the color being pink, or you can say black into white, all grey. All the offsprings are grey. Now what do we have? Now the first thing you've got to understand, there are 3 phenotypes. 1, 2, 3. If I say black into white, all black, whatever they are, then that means black is dominant. But if I say black into white and all the offsprings are white, then you know white is dominant. If I say black into white, all white, then the white is dominant. So we sometimes you get very tricky questions on this, and that is actually testing these skills, whether you know this or not. So we have codominance, and I told you in the question is that they are red into white, all pink. So this must be given in the question. Please don't, as I told you, you don't have to memorize anything. So this will be C raised to the power R, and the white will be C raised to the power W. Now you can understand why we say all will be pink. Because what is the phenomena? You have this one crossing over, this is the one which is going to fertilize this one. So we have CR, CW, and then all of them are the same. So all of them are pink. But please see how we are writing this. We're not just saying big B, small b, or big A, small p. Now we are writing C for color. And then for red I've written R. And for white I've written C, but I've written W, and this is a capital W. It's not big R, small r. So please understand that in codominance, we always use 2 letters. That is why for blood group, we use the letter IAIB. IAIB to signify that this is blood group AB. That is why I raised to the power A and I raised to the power B. That of course is decided by the Institute of Biology what letters to give. So you don't even have to know that. So this is called the phenomena of codominance. Please understand, codominance means both traits, both phenotypes, when they are both present together, then both of them show their effect. Like if you mix red paint with white paint and you get a shade of pink, or if you mix black paint and white paint and you'd get a shade of grey. So there are always, another very important thing to always remember is that there will always be 3 phenotypes. Like I said, black, white, grey. Like I said, red, white and pink. So there will always be 3 phenotypes. When you read the question, genetics questions are to be read very carefully and to be understood how you're going to handle these questions. Now, coming to the phenomena of test cross. Now, as you can see, your book says the definition of test cross is a genetic cross which an organism showing a characteristic caused by a dominant allele. So, it has to be the dominant allele. So, you have to be looking at what is in the question is the dominant allele. So, black-furred rabbits, now this is the mystery rabbit. The black-furred rabbit is either this or it is this. Well, we're not sure of this. We're not sure. Is it this or is it this? So, in order to find out what is the genotype, because we are not sure, so we cross it with a known recessive. So, you cross it with this. And I told you in the question the white fur is the recessive trait. So, all white furred rabbits will be small b. So, the main important thing to consider is that it has to be a known homozygous recessive. You have to be sure that white trait is a homozygous recessive trait. Well, there isn't any other trait. If it's recessive, it is homozygous recessive. And when you cross these with a white rabbit, because we know that white is the recessive trait, now you can see what is the possible combination. Now all the offsprings are going to be black. So we are now 100% sure that the mystery rabbit was homozygous dominant. But if this mystery rabbit was heterozygous, then what do we see? Well, we see two things. We see some with this trait and some with this trait. So this is 1:1. It's actually 2:2 because I'm not writing all of them, but it will be 2:2, or you can just simplify it and write 1:1. So please remember, in a test cross we are not sure of the dominant animal, whether the dominant animal is homozygous dominant or it is heterozygous. So in order to find that out, you have to cross it with a known recessive, with a known homozygous recessive animal. Now we are pretty sure that the white rabbits are known recessive. So when you cross these, now look at both the situation. Now this is situation 1 and this is situation 2. If it was homozygous dominant, then all the offsprings would be black. But if it was heterozygous, then you have a 1:1 chance of it being either black or white. Because you know these will be white and this will be black. So what is a test cross? A test cross is if we know that this is a dominant trait and we want to find out whether the dominant animal, is it homozygous dominant or is it heterozygous? So you will cross it with a known recessive and you will get to know the answer to it. And these of course comes in a lot of genetics questions. So you must be very aware of this if it comes in the question. Now we will discuss monohybrid and dihybrid cross. Monohybrid crosses, mono means one, so if you're only discussing one trait, like let's look at the fur of rabbits. So you're just talking of two types of fur, so black fur and white fur. So that means this is just a phenomena of dominant and recessive. One of these has to be dominant, one has to be recessive. Of course, this will be mentioned in the question. Then as compared to this, what is a dihybrid cross? Like we have disaccharides, dipeptides. So di means two, so two traits, and we're going to discuss two different characteristics. So one is the color of the stem, so they could be purple stem or they could be green stem. One would be recessive, one would be a dominant trait. And then the second trait that we are discussing is going to be the edges of the leaf. So cut leaves means that the edges have been like this, and the potato leaves are that they will have smooth edges. So we're discussing two different traits of the same plant, and these of course would be traits which would be on separate chromosomes. So this would be on one pair the gene for this would be on one pair, and this the edges of the leaves would be on another pair, and they would be somewhere here. The locus would of course be defined where it is present. So we're going to be discussing monohybrid and dihybrid cross. Now in the monohybrid cross we have three variations. Now how do we word this? This would be homozygous dominant crossed with a homozygous recessive. All the offsprings will be heterozygous. Now this is of course the F1 generation. The second variety, which is here, which I'm going to be talking about, when you cross two heterozygotes, now the offsprings would be this one and this one, and then it would be this and this, and then it would be this and this, so, and the last one would be this and this. So we have three. This would be one phenotype, and then this would be the second phenotype, but the genotype ratio would be 1:2:1. So please just remember these when whenever you have, and this would of course be a phenotype, would be 3:1 would be the phenotype ratio. Then the third one which I want you to remember is when a heterozygous is crossed with a homozygous recessive. So we have these possibilities. And this is of course 2:2 or 1:1. So these are the only ones I want you to remember of the monohybrid cross. Then we come to the dihybrid cross. Now in the dihybrid cross, as we're doing the question from the book, purple stem is capital A. So purple stem would be either capital A, or the plant would be capital A small a. The green stem would be, as it is, it's mentioned, it's a recessive trait. So green stems will always be this trait. So the cut leaves as given in the question is double D. So either big D, or it's going to be big D, small d. And that is what's given in the question, so it's not something that you need to remember. This of course is always going to be, that's also something given, the edge of the leaves is going to be small d. So in a dihybrid cross, what you've got to remember is we're talking of two traits. Now it makes very sense if you sort of understand the basic ingredient is that if it is this, and now how do you word this? Homozygous dominant for both traits. Now naturally, because so it's going to be purple, and because it's got a capital D, so it's going to be cut. So purple cut could be this, purple cut could be this. So now what we need to understand is that purple cut can be this. So it can be big A, big D, big D. It can be big A, small a, big D, small d. Or it can be big A, small a, and big D, small d. Now these are all going to be purple cut. And you can also have, so there are four possibilities. So big A, big D, big D, big A, big D, small d. And then big A, small a. So this is heterozygous, and this is heterozygous, and this is homozygous dominant. So four possibilities of having a purple stem and cut leaves. Now green stem, if we have to have a green stem, what have we have to have? If we have to have a green stem, we have to have this. That is the only possibility. So green stems will always be there. And then if you want them to be potato leaves, then they'll be this. So these will be green potato. Now in another situation in which we have small a, and big D, Now this makes sense. These will be green and these will be cut, because please remember this is cut. So what we need to understand is how are we going to cross this. So I say, okay, let's cross a heterozygous for both traits and cross it with a heterozygous for both traits. So heterozygous for the color of the stem, heterozygous for the shape, and so we cross it. Now this gives you a ratio of 9:3:3:1. Now that is always true for a dihybrid cross, because what we are thinking is that these are on two separate chromosomes. So we have big A here and we have small a here. And similarly on another chromosome we have big D and small d. So this would be in a cell. Now either they align at the equator like this, or like for instance now what is going to happen, this big A is going to go here and this big D is going to go here, and small a and small d. So we've got one possible gametes. Then in another situation what can happen is that big A can be here and small a can be here, and now they align separately. Now this is of course called independent assortment. So in this situation in a cell, this is called independent assortment, which happens at metaphase 1. So we would have big A and small d and small a and big D. So you have 4 possibilities: 1, 2, 3 and 4 possibilities. So in these 4 possibilities, then when you combine them up, you get a 16. So the 4 possibility results in 16 possible combinations. And the ratio of the combination is 9:3:1. If you add these, it's 16. So 9:3:1, and we have the possibilities here, like for instance when I wrote here A, this is heterozygous and this is also heterozygous. So you have big A, big D, so you have big A, big D here as well, big A, small d, small a, big D, and small a, small d. So the same thing you write on this side as well, and of course you complete this and you can see how it is going to result in 16 possible combinations. Now that you look at this table which has been completed for you, you can see that the 9, that's 1, 2, 3, 4, 5, 6, 7, 8 and 9. 9 are the purple cut, and 3 are the purple potato, which I've used another color for that. So 3 are the purple potato, and then the other 3 are the green cut. So we have 3 which are green cut, and then there's 1 which is green potato, which is of course both the recessive traits. So this is how we finished with the dihybrid cross, and this will continue on to video 3, which will complete the rest of this chapter, and then it will follow in the next video. This is video 3, it will go on to the video 4. Thank you.